Good morning. Really great to be kicking this off today with everybody. I'm Stef. I'm the co-founder and CEO of a company called Cradle, and I'm very excited to be talking about why I believe generative AI will have the largest impact in biology. Before starting Cradle, I was on the product leadership team of Google Research and Machine Intelligence, and a couple of years ago, we were doing a lot of work on these transformer models that are making a lot of impact right now. And this is where I got to meet my co-founder, Eli, who was one of the researchers there, and during lunch said, hey, we're applying this stuff to DNA. And I was like, DNA? Why would you want to go do that? And he said, well, maybe very similarly to how we're learning about human language, we might be able to learn about organic chemistry, and there's such an incredible amount of applications out there. That really got me incredibly excited about this space. I think I interviewed about 100 biologists to learn about their world, and I'd love to share some of the learnings of my journey with you today. So let's start with the next slide. Yes, proteins. These are proteins. And I'm going to tell you a little bit about biology first, because otherwise the rest of my presentation is not going to make a lot of sense. But this is a protein. Proteins are little machines that live inside of cells, and they do many different things. They can be a cell wall, or they can be a little machine that performs a little chemical reaction. And you can have many proteins within a cell work together to do very complicated chemistry from, let's say, a starting point like a sugar, going to a much more complicated molecule that we might want to use. We've been using that as humans for a while now. So some of you might like beer. The way that beer gets made is we throw a yeast into a fermentation tank, and it takes sugar, CO2, and water, and turns it into a more complicated molecule, which is alcohol. This is an example of one of my favorite proteins, is ATP synthase. It actually grabs little molecules around it and turns it into energy for your cell. And they can be very, very complicated machines. There's a lot of companies out there today that are building really amazing products with biology, ranging from, next slide, please. Sorry, that's too far. Yes. Some that you probably are already using. So you might have wondered at some point, why do my detergents keep working at lower temperatures? This is because people have been adding enzymes, which are a sort of protein, to the mix, because they're much better at breaking down fats, for example. And there's many really amazing companies out there that are building products using biology instead of petrochemicals or animals as an input. Let me give you a couple of examples that you might be less familiar with than, for example, let's say the Moderna vaccine. So Sologen is a great company. They're based out of Texas in the United States. They're making chemicals, but they're not making it using petro. They're not trying to use petrol as a substrate, but they're using microbes. These microbes are fed sugar, water, and CO2. So this is a chemically negative, a plant that's CO2 negative, and they turn them into precision chemicals. They can make these products cheaper than the petrochemical alternative in a way that's completely 100% sustainable. Another example is Perfect Day Foods. They're making milk, but this milk isn't been made by a cow. It's made by a fungus that's been modified to make this milk protein. And so it secretes this milk protein, and we can use it in all kinds of products. If you ever go to the Bay Area or generally to the United States, there's several products out there, including Häagen-Dazs ice cream, that is 100% chemically identical to what a cow would make, but it's made with a fungus. So 1% of the CO2 emissions, no cows were harmed in the process. Those are some of the types of companies that we are trying to enable. Next slide, please. So how do you program biology? Obviously, biology was never really meant to be programmed in the first place because it evolved. But maybe to give you a little bit of a sense of how it works, you have a sequence. A sequence is encoded in DNA, and like all of us here, lots of different proteins get made at every moment in time, and they fold into a little structure. And that structure really determines what the primary function of the protein is. And then obviously that protein also has a set of properties that biologists care about engineering. And so, for example, if you're making a therapeutic, you might want it to bind very well to a certain target, like a disease that you care about. Or in cheese making, you might want to have an enzyme that turns milk into cheese a lot faster than the ones that you might find in nature. And the way that people try to program this is by changing the sequence. Now, this is a little bit for those that are computer scientists in here, like taking binaries and then comparing different binaries and then flipping bits to see what might happen, because there's no real formal programming language for these things. Next slide, please. The way that they do this today is basically in 3 ways. One is they might use what they call a homology-based method. So they might look at what nature has done today, and they look at what are some of the safe mutations that I can make to sort of compare and contrast. The second thing that they might do is they might use these very large physics-based simulators to see how the molecule might fold and how it might interact with its environment. Or they use what they call rational design, which is basically, I did a PhD on this molecule, and I kind of know how it works by looking at it a lot. And as you're developing one of these products, and this is really the core to why I think it's really hard to program biology, is that it's really hard to make the right decision between what piece of the DNA you need to change in order to get a certain result, a certain objective met. Next slide, please. So in conventional computer programming, once you've made a change and you run your program, you get pretty quick results. And unfortunately, in biology, that is not the case, because you actually need to go try out whether the protein that you designed is better than the ones that you had before. And so the way that they do that is they'll first design this protein, so make the different types of mutations. They will then go into the laboratory and they'll order DNA. There's a couple of providers where you can just order sequences. They'll send them to you. They will put these in a cell and express, like make the proteins in the cell and then run their experiments on that. The time between, let's say, designing your candidates all the way to having an experimental result back could be 8 to 12 weeks. So if you then zoom out a little bit, when you're developing a bio-based product, you're doing many of these cycles, dozens typically, and every experiment costs around $350. And so it's quite expensive to make a bio-based product today. And that is obviously very different from writing computer software. Next slide, please. So what I've been working on with our team is very similar to some of the large language and generative models that you've seen out there today, where you can use something like ChatGPT, give it a prompt, and it will give you an answer, or generating an image where you give it a prompt and you get a really nice image. In our case, the prompt is a starting sequence. Like I have a molecule that approximately does what I want, and then your objective with it, what you would like to improve about it. And we effectively generate a set of sequences they can put into their experiments that have a much higher probability of actually reaching their design objectives. And that works better than a lot of the things that people are trying to do today when they're engineering these types of systems. Next slide, please. So this is an example of a protein that we've been working on. It's called T7 RNA polymerase. It's the little squiggly on the top left. It's a protein that's responsible for turning DNA into RNA. And so when you're making a vaccine-based therapeutic, for example, this is sort of your manufacturing workhorse that makes the actual product. You can stabilize this protein and try to make it more active, which means that it can make more RNA product, so more vaccine. And we've been running several types of lab experiments on this protein. So with some of the conventional methods that are out there, you might get 1 to 5% of your sequences coming back positively, as in being better than the ones that you had before. What we found is that with some of the generative models that we've developed, up to 95% hits on a plate, so you get a lot more sequences back that are actually a lot better than the ones that you had before. And we were able to get this thing to a target for our customer in 3 experimental rounds, where typically they might be using 8 to 10. And try to remember, it's about a quarter to run 1 experimental cycle for a lot of our customers. And so really this helps them get into the market with some of these products a lot faster. Next slide, please. So why am I excited about that? Really, because I think these types of technologies enable a lot of people that want to build products with biology a lot faster. McKinsey did some work around 60% of all of the things that we consume today, whether it's materials, foods, medicines, could be made in a fully sustainable way with help from biology. But if it costs 20, 40, several hundred millions of dollars in order to get to a working product that is too much. I think what these technologies are going to show in the coming decade is that we can build these types of bio-based products for a fraction of the amount of money and in a lot shorter amount of time than what we can do today. A lot of the venture community and sort of the technologies that I've worked on in the past were based on bits. My sense is that a lot of the future technologies and cool companies that are going to be built are based on atoms. And this is really where I think Gen AI will have some of the largest impact. Thank you very much.
Hello.
Hello.
Well done. I was just hanging out in the back watching and observing. Time for a few questions, if we may. One of the things that I thought was so compelling about this, and I'm sure I'm not the only one in this room, was what we can learn from nature in order to use AI effectively with the things you're trying to do. To what extent is nature your inspiration, or are you ignoring it?
Very much, actually. So I think maybe to give you a little bit of a sense of how you train this type of a machine learning model compared to how you train sort of a human language machine learning model, it's actually quite simple. So the way they're trained is you do an infilling task. So in this case, for example, in natural language, you would use a sentence like Stef is a bleep, and if the model says computer scientist, it gets points. If it says butcher, it doesn't get any points. If you do that for all the text on the internet, you sort of have ChatGPT 1.5, right? We do something very similar. So we take all the protein sequences that are known out there, and we do CTAG, bleep, 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 right? And then if it fills them in correctly, it gets points. And so it sort of learns what would be reasonable from an evolutionary perspective to put in a certain position. Now, that's not everything, because just a sequence might not do something. We still have to tie that to experimental results. And then we sort of expand them to some of the things that we talked about, like do molecules bind well, or are they stable molecules? And we can sort of condition these models to then also generate proteins that have these types of capabilities.
And there was a more philosophical question that I had as well. That was
you're asking an engineer, right?
Based on what you said about, you asked the question of how do you program biology, and you mentioned, of course, that biology is something that is more about evolving. So is there a tension there? Are you playing God a bit, I guess would be my question. Or are you playing Darwin?
I think it's more Darwin on steroids than God, really. Yeah,
okay.
I'm also not religious, so.
Yes.
No, but I mean, we definitely take inspiration from evolution to basically extend our capabilities into the future. I think what we do is not new, in the sense that people have been making medicines for a very long time that are not small molecules but are protein-based. We talked about detergents. Most of your detergents that you're using at home right now will likely have enzymes in them today that are modified from their sort of original one in nature. And so, it's a little bit of a sliding scale, I guess.
And are there any areas where you would feel there's a no-go area, something you're not prepared to work on and use AI in this way?
Absolutely, yeah. I mean, this is a little bit of an emerging field, especially because we're also making software. So we had to ask ourselves the question, like, what type of companies do we want to work with, and what type of companies do we not want to work with? Anybody that wants to work on things that might encode for a viral particle, or things that might be toxic to humans or carcinogenic. We actually had to figure out, like, how do you write a terms of service for this thing? And that's quite complicated. Luckily, there are a lot of controls and safeties already built into biotech today. So for example, if you order DNA from one of these providers, they will check, are you trying to be naughty here or not? And does it look like something that we wouldn't want to have out there in the community, sort of without controls on top of it? And actually, I think, maybe in contrast to people that make large language models, biologists have figured this out before they started launching technologies.
Yes, although that always leaves the possibility of unintended consequences. So how do you control for that?
Yeah, definitely. I think part of the answer is that is already also taken care of in many cases. So The area where we show up is mostly in the research and development stages, and there are very clear regulations about when you can introduce a new sort of biological into the world. So obviously in the case of medicines, you have clinical trials, and you have controls as people are building their technology in the early stages. Similarly, for things that are food products or generally sort of genetically modified systems, the people that are using our software in the R&D department, they have an obligation under law to make sure that they're compliant.
Right. And I know I've been focusing on the negative and risky side of things, but can we turn to the more hopeful side? Where do you see this going? Like, are there any things that if we could fix A, B, and C conditions, this becomes possible? Like, sketch us through what the utopian vision looks like for you.
I think for me, the utopian vision would be to replace most of the things that we're currently using animals or oil for with these types of systems. So, this is nylon. Would be nice if we could actually degrade it when we don't use it anymore, or maybe even better, we could have materials that already have the properties that make them auto-degradable. I'm excited about. I'm sort of not a science fiction guy, so I'm pretty sure people will come up with really weird applications. I got surprised, I think, a month ago when a customer signed up. They're in the world of ore sequestration, so they're in mining. I was like, mining? Why would you want to work with biology in mining? And so what they're doing is when you're trying to separate lithium from the rest of the ore, you basically need to heat it up and put a lot of very nasty chemicals inside the mix in order to get these things to separate. And they're basically making an enzyme that can do that sort of at room temperature in a bucket of water. And I was like, wow, I had never imagined somebody would do something like that. And so I think there's lots of opportunity out there. In my career, I've mostly been a tool builder, like with TensorFlow and with Android before. And so, I'm excited to see what people are going to build with this type of technique.
That's so rad. And I guess another question that I would have is for somebody who's new to this, right, who maybe is interested in AI or is interested in biology but doesn't know how to do the synthesis, how would you advise getting started and where they would begin to explore to educate themselves, besides obviously coming to CogX?
Of course, yeah. I think there's a lot of good resources out there on the internet. An organization called SynBioBeta I would definitely recommend. They have a newsletter. They have a lot of resources. Some of it is also, with every new field, like if you want to get educated about machine learning, you have to read a few books. There's a bunch of books in the biology domain. So yeah. my biology co-founder were like, here's molecular biology of the cell, read it please. And so, but Google it really is the answer.
So start with really 101. And then you're going to have to build it up.
Yeah, exactly.
And who are the companies or countries where you feel are leading? So if we had to look at the sort of global view of the field, where are you really excited about and where you see the hot spots?
You mean from a machine learning perspective or from the products
AI
that are being
biology.
made? Okay, yeah. I mean, there's in Europe, there's a couple of really great companies here. For example, Extentia, Benevolent AI, they're in the pharmaceutical domain here in the United Kingdom. Obviously, you have some more sort of general research groups like DeepMind here with AlphaFold. That's probably familiar to a lot of the folks in the audience here. I think because biotech is historically quite big in Europe, like you have a large cluster in Denmark, in the Netherlands, in Switzerland, I think a lot of these companies are starting to adapt these types of techniques, and we have a really large talent pool. So I think this is an area where we could win
right.
as Europe, which obviously for ML, we're, the center of gravity is very much in the United States, for sure.
And do you see opportunities to collaborate, particularly on like data set sharing and setting of frameworks, et cetera, or is it a very siloed space because it's so competitive?
Yeah, I think this is actually one of the complicated problems in our space. And so, if you're training something like ChatGPT, you could download the internet and sort of train on all of that data, and you have a pretty decent data set. Obviously, for a lot of people in biotech, that sequence that they're making, that's their IP, right? Sort of if they want to go sell that and they were to publish it on the internet, they just lose that IP. And so that means a lot of sort of commercially relevant experimental data and sequences are not necessarily available. This is actually a topic, I think, where we should spend more time, because good benchmarks for these types of LLMs in this space, they do exist, but they're not great. And we could definitely do a lot more there.
Wonderful. And for a bit of information before we close this session and let you run away off the stage, the Royal Society is going to be publishing a report in November in this country, so keep an eye out. They've been having roundtables with scientists. I was emceeing the one yesterday, talking about precisely this, which is why I wanted to ask. And that's going to come out with all sorts of insights, framing of problems, pointing to resources, et cetera. So if that interests you, another area to get a heads up is the Royal Society. So keep it on. All that remains is for me to thank Stef van Grieken and all of you.
Thank you very much.